Okay, I'd like to thank the organizing committee for the opportunity to share our research progress towards holistic phenotyping and understanding of the brain. I'd like to start my talk by sharing an interesting AD-related study that we did with the Liwei Tsai group at MIT. So beta amyloid, as you know, is one of the major pathogenic factors in Alzheimer's disease, and it's known to cause neurological degeneration, neurodegeneration. However, it wasn't well understood where beta amyloid plaques are formed and how the pathology progresses. To study this important question, we collaborated with the Tsai lab at MIT and created a special temporal map of the beta amyloid progression in Alzheimer's disease mouse models. The left video shows a 2-month-old mouse brain, and the right side shows a 6-month-old brain, and white dot-like aggregates in these videos are beta amyloid plaques. In 2-month-old brain, you can see that the aggregates are localized in a few brain regions, but 6-month-old brain amyloid plaques are widely spread. Through this study, we identified the early hubs where the amyloid aggregation starts, and we discovered that amyloid spreads along specific circuits. We also found that neurons in one of the hub regions show significantly higher intrinsic excitability. Interestingly, reducing activity in this brain region reduced beta amyloid pathology. There are still many important questions remaining to be answered. For example, we don't know which circuits and cell types are particularly vulnerable, even in this extensively studied mouse model. Also, considering the large evolutionary gap between mouse and human, it will be important to conduct similar studies with human brains. To study the disease mechanism and develop new therapeutic strategies for many neurological disorders, we need to study the brain at multiple scales, from the whole brain all the way down to the molecules. We also need to study the complex interactions among various molecules, cell types, circuits, and pathological factors in a holistic way other than taking reductionistic approaches. However, extracting these multi-scale molecular and anatomical information from the human brain, which is 4,000 times larger than mouse brain and extremely dense and heterogeneous, remains a major challenge. So to tackle this challenge, I have been working with a large team of incredibly talented engineers and scientists at multiple institutions listed here. And in the past 7 years, we have been working together to develop a full technology pipeline for scalable and holistic imaging and phenotyping of the human brain. Basically, we aim to develop platform technologies that allows extraction of spatial, molecular, morphological, environmental, and connectivity information all from the same brain at single cell resolution. We want the technology platform to be, we also want this technology platform to be compatible with tens of thousands of human brains currently available at hundreds of human brain banks worldwide. And the number of brains is growing. At Mass General Hospital alone, over 400 brains are donated every year. And this incredibly large number of brains cover different sex, race, age, and disease conditions. And many of them have also had rich functional and decade-long medical data associated with. These brains are arguably the most important resources for studying the neurological disorders, but they are mostly sitting on the shelves and poorly utilized. We also want our technology platform to be cost-effective and scalable so that we can routinely map many of these important brains. So to achieve this, we developed a set of technologies that engineer tissue physical chemical properties. We developed a technique called Clarity with Carl Deisler's lab at Stanford. When I was in the Carl Deisler lab at Stanford, that makes tissue optically transparent and macromolecular permeable. We developed technology called Shield that preserves all the molecular and anatomical information that we care about. We also developed a technique called MAP that expands organ scale tissue fourfold linearly so that we can do multi-scale imaging down to axonal connectivity and chemical synapses. We also developed a technique called ELAS that turns tissue into basically rubber-like hydrogel, which is stretchable and compressible. And I'll explain why we are making this transformation. Basically, using this technology, we can preserve all the key information that we care about, and we can access the preserved information. So here I'm showing how we use these technologies together to achieve phenotyping. So here is a human brain, and we developed the ultra precision vibroctome shown here to make millimeter-thick sections, thick blocks, and then we use shears to preserve all the molecules and tissue structures. And this shears platform allows us to use antibodies or mRNA fish probes to map molecules and cell types and structures that we are interested in, and we can do multiple rounds of this mapping to extract detailed information, cell type information, and molecular structure information. And all these multi-round data sets can be registered as single cell resolution. After sheared treatment, we use ELAST technique to turn this tissue into compressible and stretchable hydrogel. This video shows whole human corneal slab ELAST process, and as you can see, they are really compressible and stretchable. And with this approach, we can basically make human tissue indestructible so that we don't lose these important tissues. And all the molecular and structural information, cellular information is preserved, just like sheared. So we can use immunohistology or mRNA fish to extract out those informations. And this compression or stretching doesn't damage the tissue. So this is before image and after image showing GFAP labeled and lectin labeled blood vessels and astrocytes. And you can see all these structures are well preserved. And by combining this unique property of this ELAST with other technologies that we developed to engineer molecular interaction transport, we can significantly increase the scalability of our approach. For example, let's say we have 5 millimeter thick human brain tissue here. If you just try to stain this tissue with antibodies passively, you can only stain the surface because the antibody penetration is really slow. The dimensionless time scale can be described like this. So there is quadratic dependence between time and the length scale. So basically, if you increase the length 5 times, you increase the time required 25 times. What we can do with ELAST is we can transiently compress the tissue to shorten, basically seeing the tissue thickness. And if we do that by 6 times, we can shorten the processing time by 36 times, and we can achieve much more uniform and rapid staining. And once the tissue is stained, we can reverse the compression and then image the tissue at its native state. So using this approach, we were able to stain 5 millimeter thick human brain tissue just within 1 day, which is was 100 times faster than what we demonstrated in Clarity. So using this ELAST approach, we can preserve all the informations, and we can use low NA, low magnification objectives to image larger tissue. This is one example, human pons, brain pons, and if we zoom in 200 times, you can see individual cells. But if we want to see more detailed structures, high resolution structures like cellular morphology and axonal connectivity, then we need higher resolution. So to do higher resolution imaging of the same tissue, we combine ELAST with our technique MAP to expand the tissue. And when it's expanded, we can see individual fibers and fine morphological details of the cells. So this shows human medial temporal lobe stained with carotenoid and neurofilament antibody, and you can see individual fibers and morphological details, and you can trace them. Here's another example showing SMI31-2 labeled fibers. I will skip this. And we have been developing automated tracing algorithms to reconstruct these fibers using deep learning algorithms. And this shows fiber tracing in gray matter, and this shows fiber tracing in white matter. Okay, so using our approach, we can extract all these multi-scale informations from the same tissue in a fully integrated manner. So using our platform, in summary, we can it enables fully integrated multi-scale molecular and anatomical cell phenotyping, and it's compatible with long-banked brains and current tissue collection protocols, and it's cost-effective and scalable. And due to the time, I will skip this slide. And I want to finish my talk by acknowledging the entire team who have been making incredible progresses in the past 7 years to achieve this goal. Thank you.